Hello and welcome, everyone. In Chapter 2, How We See the Invisible World, we are going to focus on how scientists view microscopic organisms. It's going to involve the following learning objectives: understand the form and function of the parts of the microscope, how microscopy works, compare the different types of microscopy, how to observe microbes using a microscope, and staining methods for identifying and differentiating microorganisms. We will mostly focus on light microscopy because that's enough to handle in itself, and it's the most common type of microscopy. In particular, we will be looking at bright field, dark field, phase contrast, and fluorescence microscopy, although there are many other types of light microscopy. Electron microscopy is also quite common, but very technical and uses beams of electrons to look at very small objects, whether that's subcellular parts or acellular organisms like viruses. Scan probe microscopy inserts a tiny probe into a specimen and then transmits an electrical current through the probe to map the structure of the specimen. But it isn't as common as electron microscopy, where we're going to see a lot of images in throughout our slides using that. But we're going to focus on the light microscopy. This is called a compound light microscope because it uses visible light and because there are two lenses in it. Two main lens types in the compound light microscope. The first and most obvious one is the ocular lens, and this is the eyepiece which magnifies or remagnifies the image formed by the objective lens. So here at the top is the ocular lens, and here in about the midway down is our objective lens. And we're going to take you through this one at a time here. But you can look at this in another way, and you can look at this from where the path travels. So at the very here's the base of our microscope, and this is the source of illumination. It's usually just a light bulb down there at the bottom, and that light is funneled up through the it's the that light comes from what we call the illuminator, and it is funneled up through a condenser lens, and that condenser lens bends the light towards a specific point here, and that light reaches this area right here where our specimen is, or our glass slide. That's what we're trying to look at. So you can see that the light is really focused at this particular point here, and then it traverses up into our objective lens, and it goes through a series of mirrors, and this is called a prism. And what the job of the prism is just to bend that light towards our eyes so that it can come through here, the ocular lens, and reach our line of vision. We can also look at just the different parts of the microscope here. And if we take a look here at the very bottom, here's our base again. And if you look at the side here, and what isn't shown in this image is that there's two sides of this, but there's a coarse knob, which is this larger inner knob, and a fine focusing knob. These two knobs move this stage up and down, and by doing so can bring a specimen out of or into focus for the objective lens. So the lenses, they're stationary, and the stage is what moves. Again, we have our illuminator, which brings the light, a funnel of light up towards the specimen. Here we have a diaphragm here, and this diaphragm controls the amount of light that enters the condenser. So our diaphragm is kind of like the iris of your eye that can open and close. And the condenser actually funnels the light to that very specific point right at the specimen. Here is our hold, this a stage here which holds the slide. And there's typically a clip, which we can't see in this image here, but we'll go over it in our lab, which firmly holds onto your glass slide so it doesn't slip all over the place and keeps it right where you want it. To the side here, not shown very well, there's also a couple of knobs that control what we call the X and the Y axis of this stage. So this stage here, this black flat area, can move left and right and forward and backward, not up and down, but left and right and forward and backwards. So that, let's say you have this little glass slide here and you want to move it around to try and look at the left side or the right side or the top side or the back side of this glass slide. You can move it around and you're not stuck just looking in the exact center of a glass slide. Again, here's our objective lenses. And what you'll note is there are different ones here of different lengths and diameters. And those different lenses are of a different magnification. So you can actually fine tune which magnifier you want to use for your specimen. If it's a very large specimen, you may not need to zoom in as much. And if it's a very small specimen, you may need to zoom in a lot. So if you're looking at a protist, you might use a smaller magnification. And if you're looking at a bacterium, you'd want to use a high power, high magnification. There's also this part here, which is just labeled the arm. And this is where you would grab hold of the microscope. One hand along this microscope arm here, and then you'd put another hand under the base. And you always carry a microscope with two hands, never one-handed. And that would be how you would transport it. This arm is what supports the objective lenses and the prism and the ocular lenses here. The prisms are housed inside of what we call the body tube, shown here. And here again are our ocular lenses. There's two of them. And usually these ocular lenses are not only do they have a certain magnification specific to them, it's usually 4x or 10x, but they can also be swiveled around, in and out to adjust for how close or far apart each individual user's eyes are. And this part rotates as well in case one of your eyes has a different level of focus than the other eye. You can fine tune it to your left and your right eye. So now I'm going to take you through the steps of observing your specimen. And the first step here is we want to lower the stage all the way down here using the coarse adjustment knob so that we can have a cleared out area of the stage where we're not going to get, the objective lenses are not going to get in our way. You also want to rotate the nose piece. This nose piece right here allows you to switch between different magnifying objective lenses. Go ahead and switch it all the way down. Typically, you want to put it to the 4x or maybe the 10x, but usually the 4x so that you have a nice clear space there. And you're going to take your glass slide that has been prepared properly. You're going to place it on the stage and pull back this clip and slide your microscope slide at the very back here. There's a little notch for it to rest. And then if you release that clip, it's got a little arm that's going to lock that specimen into position. Now we need to raise the stage back up using the coarse adjustment knob. Again, that's down here. It's a larger inner knob. You're going to raise it up slowly while looking through your ocular lens, patiently waiting until you bring that glass slide into focus. And I'm going to have an extra step, which I'm going to show you in lab. But bringing it up into focus, we just keep this nice and simple. We're ready to then move on to the next step. And that is to switch to the next magnification setting up. And from there on out, because we're working with such minute changes in the stage, you want to only use the fine focusing knob. If you use that coarse knob, if you touch it, you're going to bring it so far out of focus that you might not know where to go to get it back in focus again, believe it or not. Then after you get it into focus, you're ready to repeat these steps again for the next magnification, or 40x on most microscopes. And once we're ready to move up to the highest power magnification, let's say you're looking at a bacterium and you need to get to 100x, you're going to want to rotate up to the 100x with an extra step. So halfway between the 40x objective lens and the 100x objective lens, you're going to turn it, and that will allow you to have some space right above the glass lens where there's no objective lens hanging over the top directly, some space for you to grab some immersion oil, is what we call it, or lens oil. And you're going to add a couple of drops. There's usually just a glass dropper, and you just gently touch the glass slide with the oil and apply 1 to 2 drops of oil, not too much. You don't want to make too much of a mess here. And then you'll complete the rotation up to the highest magnification. And then you're going to continue with your magnification. And once you get your specimen in focus there, feel free to use the X and Y control knobs that I told you about that are going to let you move the stage from left to right or forward and backward to scan around on that image there and look at what you need to look at. So, why is it that we add immersion oil? Well, when we think about the way that the microscope is designed, if we recall, we have this diaphragm here that opens and closes a giant circle that determines the diameter of the light that's allowed to enter. Anything that's this wide can enter into the microscope. Into the condenser, and this can also narrow and be made smaller, so a smaller diameter of light lets in. And above that is we have this condenser here that's going to direct the light towards your specimen right here, pinpointed in the middle. And these glass lenses have, because of the shape of them, when light hits glass, a solid, it bends the light towards the angle of that glass. And as we traverse through these glass condenser lenses, they don't magnify like an objective lens, they just curve the light over. What we get is light that has a very specific angle. And when it passes again through your glass slide into air, if we look at the red here, that glass slide is going to bend the light again. And it will bend the light in such a manner that it will bend it away from your objective lens at 100x. And we want to stop that. This process is what we call, this phenomenon is called the refractive index, or how much light will bend as it passes through a particular material, whether that's the glass or the air or immersion oil. Oil is a wonderful material to use in microscopy because it has the same refractive index as glass does. And so if we add a couple of drops of oil, switch to that 100x, and that oil can bridge the gap so that there's no air bubbles between the glass slide and your objective lens, it's going to prevent that curving of the light path away from your objective lens and complete that continuous chain so that it reaches your objective lens and travels up into the prism and reaches your eye. Okay, so now that we know how the different parts of the microscope, how to use a microscope, and what immersion oil is and refractive index is, let's talk a little bit about total magnification. Total magnification is going to be how much magnification you get of your specimen above what you can just see with your unaided eye. And it's a combination of both lenses, the objective lens power and the ocular lens power. What we mean by power is how many times bigger does it make the object. So if you're looking through a 4x objective lens, you're going to actually increase the size of that object by 4 times. And let's say that your ocular lens is 10x on the microscope, although each microscope can be different in its ocular lens magnification. It will say on the side somewhere, it'll say a number and an X to tell you what size it is. If that ocular lens is also 10x, then we would simply multiply the two together. So 4 times 10 means that our total magnification is going to be 40 times bigger than what it looks like with the unaided eye. Finally, let's discuss resolution. Resolution is a very important topic, and it's what makes really good microscopic images. Resolution is by definition, the ability to distinguish between two separate points. So, depending on what device you're using right now, you may or may not have the resolution to distinguish between the two periods I have written on the slide here, typed on the slide here, and it's circled in red there. And hopefully, your resolution allows you to tell the difference between those two dots. The higher the resolution you have, the smaller the space between those two dots can be, but you can still differentiate the two. Ultimately, what this means is that the lower resolution you have a fuzzier image, and the higher the resolution, the sharper the image is. And those there are two factors that can affect this. The first being wavelength of the light. Light rays can come in different wavelengths, and we'll show that here on the next slide. And the shorter the wavelength is, the more capable you will be able to determine if there's that one period there or two periods there. The second factor is the numerical aperture. We've already discussed magnification, which is the ability to increase in size compared to the real size of an object against the unaided eye. Numerical aperture is the lens's physical ability to gather light. And we're not going to get into the mathematics of that, but the more light that it can gather, the better. So with numerical aperture, the higher the numerical aperture, the better for resolution. Let's return to wavelengths. What do we mean when we say the wavelength of light? Each type of light has a certain features about it, and here's a cartoon depiction of a wavelength of light. We can see here's the wave, and the waves have both a crest, which is the top point, and a trough, which is the bottom point of a wavelength of light. And the distance between a crest to a crest, or even a trough to a trough, they're the same in all intents and purposes. That is what we define as the wavelength. And the wavelength actually makes a huge difference when magnifying an object. The shorter these waves are, the higher the resolution we can see, because these shorter waves they have less overlap, and they blend with other light traveling in the same direction. The longer the waves, the more they overlap with one another, and the more blurry an image appears. So we want nice short wavelengths in order to have a better resolution. What we call light in our day-to-day conversations is really just a portion of light that humans can observe, visible light spectrum. Another name for light is electromagnetic particles. As humans, we can only see what we call the visible light portion of the scale. In this scale, blue light is the shortest of all the wavelengths. So by using bluer light as an illumination source, the resolution can be mildly improved. This is a limitation of light microscopy. Resolution will be no better than the visible light spectrum, or down to, as we can see, about 380 nanometers or 0.38 micrometers, where our vision ends. And you can see that in the image above. Can you recall how small bacteria are? Somewhere around 1 to 2 micrometers. And how small are viruses? Generally 0.3 micrometers or smaller. Therefore, you can see that even with our best magnification, that makes it pretty difficult to see much detail with bacteria. And we can't see anything at all when it comes to viruses. So when we observe images under the microscope, it's a much different ballgame than what we would like to see in some of these electron microscope slides. Images. Our maximum magnification using bright field light microscopes in most labs is going to be about 100x for the objective lens and 10x for the ocular lens, or about 1,000 times magnification. And that's going to be about 1 micrometer, about the size of a bacterium. Let's now talk about the different types of light microscopes that are out there. The first one and the simplest one is the bright field microscope that uses visible light to illuminate a specimen. Imagine the light being filtered through the diaphragm through to the condenser and onto your specimen. The specimen is going to block out or shade some of the light. And anywhere there isn't a specimen or an organism usually present, or no objects present on that glass slide, there will be no shading and it will appear bright. So the specimen is actually a darker image surrounded by a bright background, or in other words, a bright field. The resolution limit here again, if you're amazingly good with your resolution, you have a really good microscope with a good blue source of light and a high numerical aperture, is allegedly around zero point 0.2 micrometers. Here we have a large paramecium, a protist, that is about 100 times as wide as a bacterium. In practice, zero point 0.2 micrometers is going to leave us wanting a lot more. And it's not going to give us a lot of information about some of the intracellular details of an organism. Dark field microscopy is a type of light microscopy that contrasts sharply with bright field microscopy. In dark field microscopy, a specialized condenser is used to block direct light from reaching the specimen. This condenser includes an opaque disc that prevents central light rays from passing through, allowing only oblique light on the sides there to illuminate the sample. The key principle is that only light scattered, or we call that refracted, by the specimen enters the objective lens, while unscattered light is excluded. As a result, the background appears dark and the specimen appears bright against it. This technique is particularly effective for observing small, transparent, or unstained specimens that are difficult to see using bright field microscopy. The, the opaque disc positioned close to the light source ensures that most of the light is blocked. The remaining light is condensed and directed around the edges of the specimen. This oblique light is then refracted or reflected by the specimen into the objective lens, creating the bright image of the specimen against a dark background. Dark field microscopy emphasizes the edges and structures of the specimen, making it an excellent tool for visualizing fine detail without the needs for staining. As a result, the image is inverted and only the specimen is illuminated. This is really useful for visualizing organisms that can't be seen using bright field microscopy, typically those that don't stain very well or those that might have to be kept live while observing them, and you don't want to destroy them in the staining process. Um, Brightening up only the specimen helps to highlight it when staining cannot be done. Here's an example of a bright field, of a dark field microscopy image of a paramecium. And if we go ahead and take a look at the scale bar here, we will see that the length of it's a length of about 25 micrometers in the scale. So this is probably about 6 to 9 times that scale bar. This would confirm about the right size of a paramecium of about 200 micrometers. Something really beautiful about dark field microscopy is that we can see the different parts of what is called the ultrastructure of the cell, or the inner parts of this cell. So there's some organelles and things we are able to see inside of the cell that we weren't able to see with that bright field microscopy image. Phase contrast is a very complicated form of microscopy. It works by combining two types of light that enter the objective lens: direct light from the light source and light that is reflected or diffracted by the specimen itself. The microscope uses a special phase plate to manipulate the light waves. It shifts the phase of which we'll talk about in a moment, of the direct light slightly compared to the diffracted light, which enhances the interference between the two. This interference amplifies subtle differences in light intensity, making variations in the thickness or density of the specimen more visible. Generally, there's no need to fix or stain specimens with phase contrast microscopy, and it provides very deep visualizations of the specimen. This technique allows for much better visualization of internal cellular structures as well. You can see that these internal structures have much more contrast and tend to pop out a lot better compared to bright field or dark field microscopy. We're not going to get into too much detail about how phase contrast microscopes work, but suffice it to say that by manipulating the phase of light waves, the microscope amplifies this image, creating a deeper and richer visualization. So, what do I mean by altering the phases and phase contrast? Well, imagine waves in an ocean. If two waves are moving in the same direction and they come together and overlap each other, they can cancel each other out or make each other stronger. If you look at the image here, if the waves are both going up and down at the same time at the top there, they reinforce each other and lead to what is called constructive interference. And what happens is the height of the crest or the trough get amplified. If one wave, however, is going up and the other wave is going down and they are combined together, they cancel each other out. And this is called destructive interference. The same idea applies to light waves. Remember that light refracts as it passes through an object. In typical bright field microscopy, this refracted light creates the shadowing because it doesn't reach the objective lens, and so it's removed from the image. In phase contrast microscopy, the refracted light and direct light are carefully manipulated to interact with each other. A special phase plate shifts the light waves so they are combined together and overlap just the right way to create either constructive or destructive interference. We won't bother going into any more depth on the mechanism, but you just need to be aware of the basic function of constructive and deconstructive interference, and this is what causes the richness of the image. Another fun type of microscopy is fluorescence microscopy. And this again uses visible light, sometimes even all the way to those really short wavelengths, which are called ultraviolet or UV light. So that would be a little bit smaller of a wavelength than the violet we're able to pick up with our own eyes. It utilizes fluorescence, which is the absorption of light by a substance, typically at a shorter wavelength, and results in that substance emitting its own light at a different color of light at a longer wavelength. And if you need to refer back to the scale I showed you of the different visible light waves, please do. But violet is kind of on that far left side and it has the very short wavelengths, and all the other colors you can see are going to be longer wavelength all the way over to far red. Fluorescence microscopy makes these very brilliant colored images. And the way it does this is it uses either naturally fluorescent organisms or organisms that are stained with what we call fluorochromes. These are fluorescent dyes that attach to structures in the cell or on the cell. In our example, ultraviolet light will stimulate the fluorochromes and cause them to get excited. And then when they get excited, they release their own light, similar to how the fluorochromes found in glow-in-the-dark toys work. A filter is then placed between the specimen and the ocular lens that blocks the light used to stimulate the fluorochrome and only allows the light emitted from the samples itself, so the fluorochromes, to pass through. And this causes the specimen to appear as a brilliant color of whatever emitted light that was against a dark background because our illumination source, say ultraviolet, has been filtered out with that filter. Fluorescence microscopy has specialty purposes. Certain species of bacteria can be effectively stained and detected with this technique. And this image is from the OpenStax textbook and is a fluorescent stain used to visualize larvae of Schistosoma mannsoni. which is a parasitic worm that causes intestinal diseases, which are common in the tropics. They are likely being exposed to a UV light and fluorescing this brilliant green color, and a UV filter is applied to the specimen and objective lens so that the specimen is more easily observable and the observer's eyes are not exposed to any harmful UV light. Here we can see that ultraviolet light is a shorter wavelength, and when a fluorochrome that is sensitive to UV light is stimulated with it, then it will release one of these longer wavelengths of light. And we most commonly see this as a green light. One example of a fluorescing protein found in nature is what we call green fluorescent protein, or GFP. It's a very common fluorescent marker. It's coded by a very small gene of only about 300 base pairs in length, which in terms of genes is about half the size of the average gene. It's often used as what we call a tag for bacterium. So the image you see off to the right, I genetically engineered or tagged this cyanobacterium to make the GFP protein. This is something very similar to what you all did in your final lab in Bio 181 if you took it at CAC, where you introduced GFP into E. coli. So in this image, I exposed the bacterium with UV light, and the specimen gave off this brilliant green fluorescent color. And again, we had a filter that prevented any UV light passing, and all you can see is that bright green color coming from the bacterium themselves. Here is another application of fluorescence microscopy. The image right here, Candidatus prometheoarchaeum syntrophicum, is closely related to archaea, bacteria, and eukarya. What that means is that we're really close to the base of the phylogenetic tree of life, where the different domains of life diverged. There's actually a culture of many organisms here, all working together in a process called symbiosis. The presence of these other organisms, and each one of them, is very hard to see because they're tiny bacteria and they are clustered together and they can't live in existence without each other working together. However, by using fluorescence microscopy, we can detect fluorescent signals from each of the different types of bacteria. And we can apply a computer software program to count those individual particles based upon their color, or count those individual bacteria in order to ascertain the total size of their population within the culture. In this way, scientists were able to observe which microbes were working together in this community through their cohabitation. And without the color, we would probably do a poor job at identifying which of these little small dots represents which bacteria, again, which are very small for doing light microscopy. But since each of them has their own unique fluorescent signature, it becomes possible to process and quantify their numbers. And that brings us to our discussion of how samples of microbes can be prepared for light microscopy. And that brings us to staining and smearing, something that your parents never wanted you to do, but we're going to do a lot of in this class. Well, staining is a way of coloring a specimen with a specific dye that emphasizes certain structures. However, before staining a specimen, it must first be attached to our glass slide. These specimens are almost always in a culture of many microbes, and the specific term for these broth cultures is called liquid media. By creating a thin layer of this liquid culture on the glass slide, which is called a smear, and then making sure it is attached well or fixed to the slide so it doesn't come loose during processing or our staining process for examination under the microscope, we can then begin the process of staining. The steps of staining begin at the top of this image. It can be done from liquid cultures, as I described, on the left side shown in the image, or cultures grown on solid nutrients, like a petri dish, which you've used in a prior biology, such as Bio 181 class. The culture is spread with an inoculation loop from liquid media by dipping the loop in the liquid broth. Much like a bubble wand, you would dip into the liquid in order to blow bubbles. You're going to dip that inoculation loop into the liquid, and it's going to form a very thin film across that loop that can then be dabbed onto the slide and smeared around into a very thin, small circle. A quick examination under some light should show a very, very faint smear. Again, we don't want to have too thick of a smear, or else bacteria will lay on top of each other and block out the light for each other. So we must avoid having that too thick of a layer there, so that we can get a clear image of each one of those microbes. For solid media, it's a little bit different. In other words, these are microbes that are grown on top of plates, as we've discussed, or in sometimes we have tubes that we grow them in with solid medium in them as well. And what we want to use instead of a loop is a needle. So you should see off to the right side here. Here's a very small needle, and you use that needle to just grab a very small amount of bacteria and smear them around as well. And now we're ready to dry that culture onto that glass slide. And this is part of the process of fixing those microbes to that slide. The first thing you want to do is just allow it to air dry in the room. It's very thin, so it shouldn't take long for it to dry. We don't want residual water on the slide, because the next step we're going to do is apply some heat. And the heat makes it kind of really fix against the slide material. And if there's still water there, and we put that water underneath, say, a Bunsen burner or an incinerator, it can cause that water to boil off rapidly, and that can actually cause damage to your cells. So what you want to do is just let it air dry a little bit. And then if you have a heating element such as an incinerator, you're going to hold it against the sidewall of it for maybe about 20 seconds until it kind of warms up a little bit. You don't want to get it too hot there, but about 20 seconds is pretty good. If you have a Bunsen burner here in a flame, you will use some pair of forceps or hemostats or something like that, and just quickly pass it through the flame a couple of times, let it rest, and to do maybe a couple quick passes again. And it's a little bit tricky, but you should get the hang of it pretty soon. Now, let's move along to the stain component now that our slides are fixed. Stains are composed of different chemicals. Stains are salts. A salt is a chemical composed of one part of a positive group and one part of a negatively charged group that are attracted together and form a salt. Kind of like sodium chloride or table salt. You have sodium, which is positively charged, and chloride, which is negatively charged. And that positive and negatively charged ion attracts them to each other, and that's what keeps them stuck together. So stains are composed of a positive and negative ion, and you might want to do a little bit of a review of what those are. Essentially, negative ions are molecules or chemicals or elements or atoms that have an excess of electrons, and electrons are negatively charged, and so they make it a negatively charged ion, and vice versa. A positively charged ion has fewer electrons than protons, and protons are the positively charged components of a element. And so if you have less electrons to balance out the protons, it becomes positively charged. We have two categories of stains, and those are going to be a basic dye. And a basic dye is when the colored portion, called the chromophore, is the positive ion. And a acidic dye is when that chromophore is the negative ion. Basic dyes are going to be the most common type of dye that you use. Why? Because the chromophore, being positively charged, is going to like to stick to the microbes. Most microorganisms have a lot of negative charges on the outside surface of the cell, and therefore that dye is going to love to stick to that organism. We can see in the image here to the left, that's a basic dye, and we have a positive staining of those organisms. And with an acidic dye, that chromophore actually is going to be repelled by those negatively charged surfaces of that microorganism. And so they're going to appear as a stainless object within a stained background. There are three different kinds of staining techniques that we can do here. The first and most simplest one is simple staining. It uses just a single basic dye. And remember, basic dyes are going to attach to the outside of the cell. And this is going to highlight the entire microorganism and allows us to visualize the cell shape and sometimes some internal structures. In the example you can see here, this is methylene blue, a very common basic dye that we use in the lab. And we've stained human cheek cells here. So simple staining is just a single, just one basic dye added to your slide. As you can see, the cells are colored and the background is not. And so this is going to be only good. Simple staining is good if you already know what you have under the microscope and you just need to visualize them for analysis. So if we're trying to just see our cheek cells and we take a swab of them, we don't need to use any other complex staining here. We can just use a simple basic dye here. Now, differential staining involves identifying an organism from possibly other organisms. Differential stains tend to be much more complex because they involve multiple dyes, at least two. What makes it a differential stain is that it is used to distinguish between two types of cells, typically bacterial cells. The most common of which are gram stains and acid-fast stains. In the example image, you can see this is a gram stain of Lactobacillus crispatus. And it's purple, so it is what we call a gram-positive stain. The purple color makes it a gram-positive stain, which we'll discuss on the next slide. And again, and make sure you're always capitaling the gram and gram stain. This is out of the textbook here, but gram, this stain is named after a person, and so it should be capitalized. Again, the gram stain is by far the most common stain used in microbiology. You will need to memorize each and every step in the staining process. The chemical agents change between different differential stains, but there are many similarities, and here are the general steps in the gram stain. Step number one is the addition of the primary stain in the gram stain. This is crystal violet. Crystal violet dyes all of the cells a purple color, regardless of them being gram negative or gram positive. In this first step, so basically all cells should be purple after this step. The second step is the addition of a mordant. A mordant is an ion that binds to the dye and helps it to hold itself in place better. Sometimes we might even use actually just heat as a mordant, as in the acid-fast stain, which will come later. In this example, it's a chemical mordant called iodine. This creates what we call the crystal violet iodine complex. The next step in the process is to get rid of the primary stain from one type of cell so that we can apply our contrasting stain to those cells. Here we have an alcohol wash. We apply alcohol for just a few seconds in order to cause the decolorizing to occur to those that are gram-negative cells. Those gram-negative cells will lose their dye readily. However, the gram-positive cells will retain their dye. So at this point, you should have either purple gram-positive cells or colorless gram-negative cells. And then lastly, our secondary stain. We will add a secondary stain so that we don't just have colorless cells. If there are gram-negative cells present, we can still observe them. And that counter stain is called safranin in the gram stain. And this is the color of pink. So basically, we have step 1 through 4, and at the end of this, we should have purple or dark blue color for gram-positive cells. We can see here our coccus cells are gram-positive, and a pink rod shape is our gram-negatives in this example. If we zoom in and look a little bit closer to actual samples, here we'll see we have two different types of bacterium. And it's obvious because one of them is coccus shaped and the other one is rod shaped or bacillus shaped. But you should also be able to differentiate that the caucus ones are that dark blue or purplish color sometimes, and that indicates that it's gram positive cells, whereas the gram negatives are that pinkish color, making it a gram negative cell. But why do some bacteria stain gram positive and others stain gram negative, and why are we so concerned with this? Well, this actually gets to the thickness of their peptidoglycan layer. Organisms that have a very thick peptidoglycan layer, gram positive organisms, are very good at retaining that crystal violet iodine complex, whereas organisms with very thin peptidoglycan layers are very easily destained using our alcohol wash. That's because the alcohol more easily penetrates into the organism and that stain is more easily released. And so, using this method here, we can tell the difference between gram positive and gram negative organisms, making it very easy to broadly categorize these organisms. But also, peptidoglycan is one of those components in bacterium that we use antibiotics to attack. And so, knowing what type of organism you have based upon its peptidoglycan layer can help you quickly determine what sort of antibiotics might be useful for fighting off an infection. The next stain we're going to discuss briefly is the acid-fast stains. Acid-fast stains work on two types of bacterium that have waxy material in their cells. This includes bacteria from the genus Mycobacterium, which are bacterium such as M. tuberculosis, which causes tuberculosis, and M. leprae, which causes leprosy, and also nocardia, some of which are non pathogenic and others are weakly pathogenic to those who are immunocompromised. So, this stain is very important for screening for these organisms in the medical field. This is how the acid fast stain is conducted. Once again, it's going to be very similar to the gram stain that we discussed, and you're going to begin with a primary stain, which is first added. Instead of crystal violet, this process uses carbonyl fusion, which is applied to a fixed smear of cells, and it colors all of the cells pink, not purple. Next, heat is used as the mordant, not iodine, but heat. So the slide is gently heated over a small heater or water bath to prevent it from drying out during the mordant process. Then we use a decolorizer. To differentiate, we need to remove the primary stain where it doesn't belong. In this case, an acid alcohol decolorizer is used. And then finally, the counter stain is applied. In an acid fast stain, the counter stain is methylene blue. We showed methylene blue earlier when we stained the cheek cells in our simple stain. So this is going to stain all of the cells that don't have a waxy cell wall. If they don't have a waxy cell wall, they easily are going to lose the carbonyl fusion during the acid alcohol decolorization step, and then they'll be able to take up the methylene blue. A couple important things to note here is this process, again, is kind of the opposite in colorization to a gram stain. If a organism is positive for the acid fast stain, they're going to be a pinkish color, and if they're negative for the acid fast stain, then they should be a bluish color. Some microbes can contain a protective coating, which we call the capsule. Capsules are important to many pathogenic bacterium because it protects them from harm by the immune system. The capsule can indicate the degree of violence that a pathogen might have, which is the ability to cause disease. These capsules do not take up most dyes, and they appear as an unstained halo that surrounds the cell when stained with India ink or another stain called negrosin. This is a type of special staining that is done, and it's referred to as a negative staining because the background is stained and the organ of the cell wall, the organism or the cell wall itself is unstained. The next stain is endospore staining. Endospores are another structure by the cells. You could think of endospores as sort of like bomb shelters for bacteria, where they can hunker down and survive rough times. These bomb shelters are consequently also very difficult to stain, but it can be done using a lot of heat and time to weaken the structure and allow the dye to absorb. Endospores arise in certain bacteria under a stressful environmental condition. The staining requires the Schaefer-Fulton method. The primary stain is malachite green, and it uses a heat source to penetrate the endospore, and the decolorizer is just water and a counterstain of safranin. The consequence of which, as you can see in the image, is that the endospore sustains a green color from the malachite green, which is held during the decolorization and counterstaining process. And the counterstain pink should dye cells that are not endospores a pinkish color. The last stain in our discussion is flagella staining. Flagella are very small appendages attached to the outside of microbes, and they often aid in motility. Most bacteria are only, again, about 1 to 2 micrometers wide. And so these thin appendages coming off of them are even more extremely difficult to see with the resolution of a light microscope. An unstained flagellum is so small that it just can't even be visualized typically with a light microscope. So we have a workaround with this. If you use carbonyl fusion and a mordant, you can apply a layer of thick dye onto the flagella. This process coats the flagella with enough stain to make them visible under light microscopy. And that's going to complete our discussion of how microbiologists visualize the microscopic world. Of course, this work is made up of many disciplines, but this should suffice as a good primer. Thank you all for joining me, and I hope you all enjoyed.